Hi everyone, I'm Cassidy Weavers and I'm Cassidy Weavers, and my summer scholarship was using a novel method for parallel taxonomic sequencing and also developing this method to work better for us. I started this project from some work that had already been done by the Remus Emerson lab group. They went out last year and sampled 5 Arabidopsis thaliana plants out in the environment, and they took many samples, bacterial samples from these plants, and stocked 202 strains, isolated strains, into glycerol. And here you can see some of the diversity that's present on these plants. And I started from here. So I grew each of these strains on agar, extracted their DNA to run a PCR targeting their full ribosomal operon, and then running the full 96 well plates worth of in a library on a Oxford nanopore flongle. We can run full 96 well plates on one single run in parallel due to the unique primer pairs that we have combined. So we have eight unique forward primers that bind to the 16S RNA operon and 12 reverse primers that bind to the 23S RNA operon. And so there's the portion of the primer that binds to the operon, and then a portion that is an adapter. And as you can see here, this is the part that binds to the ribosomal operon, and then this is the adapter, and that way we end up with 2 unique adapters on the end of each sample. And later we can use our results from the flongle and pull apart each individual sample by using a bioinformatics pipeline. And as we know, nothing in science ever works the first time around. So there's many more steps in that last slide than what I put on. So far I have poured many plates, run about 644 individual PCRs, 11 gels that most of those have full 96 well plates on, and we've done 1 flongle run last week. And I've probably done this a few million times. So this is one of the graphs resulting from our first flongle run, and we can see that so the full ribosomal operon is 4,500 base pairs, which has a high read count that we can see here. There's also a high read count here in the much lower, shorter fragments, but this is because shorter fragments are read more frequently through the pores on the flongle. And if we switched this to the base read version of this graph, we can see that our targeted sequence is much higher read and gives us what we want. And so we're still in the process of making our way through this, but so far from what we've seen, there are many bacterial strains present that we would expect to see. And so why is this technique useful? Using this technique, we can do high resolution taxonomy by using the entire ribosomal operon rather than just the traditional 16S RNA operon. It's high throughput, as I've mentioned, for running entire 96 sample libraries on the flongle in one go. And because of this, it's also cost effective. So a flongle is $90 US, which works out to about 95 US cents per sequence that we get out of it. And we were only limited to 96 sequences in this instance because of our 20 unique primers. So theoretically, with more unique primers, we can run many more samples in a single library, and also longer sequences if needed as well, because the step up from the flongle is the min-ion, and that runs 10 times the size or length. And thank you very much to the BPRC and the Remus Emerson lab for this opportunity, and I've had a great time.
questions for Cassidy?
I mean, is there much recombination in the operon, or is it effectively like just one very data-rich sequence that's not in the
So it's from the 16S operand all the way through with the spacer through the 23S. And so that gives us a really detailed taxonomic identification rather than just using this. And because there's lots, that we're even finding that there's lots of alterations in just the 16S that would sometimes identify something as a different species, but it's actually still the same species. And this method can have, it's got insertion deletion issues sometimes, and so using a longer sequence, we have higher resolution and more certainty.
What about on the run you've done already? Have you got all possible combinations back?
So we've just yesterday we were figuring out the best way to put in the adapter and primer sequences for the pipeline to like spit out them all individually, but we have done some manually and we've got quite a few. So this first plate that has been run on the Flongle is many fast growing strains, so lots of Pseudomonas and things that you would expect to see. And we're also using this later on. It would be great for community sequencing using unique primers, and then you can find the community makeup on plants.
Thank you very much.